Welcome to the campus of North Idaho College and today's broadcast of the North Idaho College Public Forum. The crew is comprised of North Idaho College television students. Your moderator is North Idaho College political scientist Tony Stewart.
Welcome to our fifth program in a 6-week series on the subject of DNA research and genetic engineering. On the previous programs, as you well know, we've been dealing with the ethical questions of this research and, secondly, the legal complications and the issue of public policy. Today and next week, it is our very special privilege to welcome to the program a scientist who will deal with the technical issues and processes that are involved in DNA research and genetic engineering. A very distinguished guest is with us today, Dr. Martin Klein, who is a professor of medicine at the School of Medicine at UCLA. Dr. Klein holds a baccalaureate degree from the University of Pennsylvania and received his medical degree from Harvard University. In addition to that, he is well published with over 250 articles and lectures. He is the author of 7 books, including his most recent publication of this year, which is a new textbook of internal medicine. Dr. Klein, as you can well observe from the October 20th issue of 1980 of Time magazine, is probably best known for his research dealing with the inserting of foreign genes into the bone marrow cells of mice and recently in Israel into 2 women. We're going to be discussing that and other things for the next half hour. Dr. Klein, welcome to our program.
Thank you.
Joining with me in questioning our guest are 4 members of the panel. First of all is Mr. Bob Kabler, who is chairman of the Department of Physical Sciences and Mathematics at North Idaho College, Don Sprague in the Department of Psychology, Richard Snyder in the Department of Anthropology, and Father Bill Wasmuth of the St. Pius Catholic Church in Coeur d'Alene. Gentlemen, welcome. I will invite Bob Kabler to commence the questioning.
Dr. Klein, perhaps you could begin by just giving us a brief overview of what is meant by gene transfer or gene transplant techniques and what the research in the area is at right now.
All right, it might be useful to start with explaining what a gene is and then going from that point. A gene is a basic unit of heredity. In all animals, plants, and man, genes determine how we look and basically, potentially, how we act. They determine skin color, hair color. Complexes of genes determine height, and so forth. Beginning in the early 1970s, a new branch of science, of biology, arose in which experimentalists were first able to isolate genes and to combine them, to splice them together. This type of research has been called gene splicing research, sometimes called genetic engineering. It's sometimes called recombinant DNA research. Recombinant DNA because one recombines genes from one species, say a germ, and another species, say a mouse. Genes are made up of complex organic molecules. The whole complex is called DNA, which is a shorthand. And gene splicing techniques involve the splicing or joining of 2 different DNAs together. The first applications of this type of new biology to medicine has been in the production of hormones like insulin, which are used to treat diabetes. Scientists have been able to identify the gene that makes insulin. They've been able to, in essence, construct an artificial gene for insulin, and by inserting that gene into bacteria, have been able, in essence, to teach the bacteria to make insulin. In this case, teach bacteria to make human insulin, which has now been used to treat patients with diabetes. And this is now a new emerging field of technology in which one can engineer bacteria to make products that are potentially useful for treating human diseases. Products such as insulin or other hormones or vaccines, which are very difficult or impossibly expensive to make by other means. That's one type of application of genetic engineering, the insertion of genes into bacteria to have them make a product. A second type of engineering, which is just now beginning, whose history is only 3 or 4 years old, is the insertion of genes into animal cells. At first, these techniques have been applied to cells from animals which grow continuously outside the body under well-defined conditions. The cells will grow in mixtures of serum from animals and of salt solutions, and will continue to divide essentially indefinitely. They're said to be immortalized. And scientists have been able, by some very ingenious strategies, to insert well-defined genes into such cells growing in culture. And within the last 2 years, attempts have been made to transplant this technology from cells in culture to cells of living animals. And mice have been the species of animal which has been examined.
Mr. Sprague.
Okay, thank you. Dr. Klein, you made a big move. You moved from the experimental lab to trying to apply this with human subjects, and you've come under a fair amount of fire, not only from the popular media, but also from your colleagues within the field. And in an article from Science magazine dated 31 October 1980, Richard Axel of the Columbia College of Physicians and Surgeons says, "There is simply no scientific basis for expecting this experiment to work in people. A lot more has to be experimented with in animal systems. Klein has done this experiment in a mouse, and as I understand it, it didn't work. He has made a great conceptual leap from a failure in a model system to trying it in humans. He is saying, it didn't work in mice, so I'm going to try it in man." Were you surprised at this very negative reaction from other scientists? And how do you respond to these individuals?
Well, yes, I was surprised, because if that statement is accurately quoted, Dr. Axel would have no way of knowing what all the antecedent experiments in animals were, since only a small fraction of the animal studies have been published. It might be worth just telling you what has been done in animals so that you and the audience can understand. What my colleagues and I attempted to do beginning over 2 years ago was to insert new, well-defined single genes into the bone marrow cells of mice. Bone marrow is where blood cells are normally produced, red blood cells, white blood cells, and so forth. And we based our studies on a series of preceding studies that had been done in tissue culture by Axel, actually, and people in his group, as well as other scientists. Techniques whereby one could take a purified cloned gene and insert it into a tissue culture cell. And by cloned, I mean a gene in which one makes thousands or millions of identical copies. Well, we took the strategy that had been used in tissue culture and applied it to the bone marrow cells of living animals. We, in essence, inserted new genes into these cells and restored the cells to the mice, where the cells divided and formed blood cells, and in addition expressed newly inserted genes. And we had experience with literally hundreds of mice with these experiments over a period of several years. And we'd learned a good deal about the most efficient way of introducing genes, something about how they're expressed, and also a chance to observe whether there was any potential harmful effects on the animals. We've observed hundreds of animals, as I said, and some of these animals we've observed for nearly their entire lifespan while they carry new genes, and observed no toxicity in the animal. We've had very clear evidence of success with at least two genes in the mouse studies. After a good deal of consideration, the time seemed ripe to begin to apply this type of information to patients who were suffering from otherwise fatal diseases. Patients whose life expectancy was measured in terms of a year or 2 years. In the particular disease which we chose to begin, a type of anemia that's common around the Mediterranean basin, life expectancy is only up to the late teens or early 20s. It's a fatal illness. We anticipated that it will take many years to perfect gene therapy of lethal inherited diseases. The time frame for success might well be as long as a decade. My colleagues and I felt that we were ready to begin such studies. The potentials for doing harm were very small. There's a good deal of information on that point. And although the chances of success, complete success at the beginning, were relatively small, notwithstanding there was such a chance, and we intended to apply the techniques to patients with otherwise fatal illnesses. And so we devised a long-term strategy that would go over a number of years, in which we would sequentially examine the success or failure of our experiments.
Ladies and gentlemen, if you've joined our program in progress, our guest is Dr. Martin Klein, speaking with us in our 6-week series concerning genetic engineering for the purpose of trying to cure human diseases. We shall continue the questioning with Richard Snyder.
Dr. Klein, as an anthropologist, I'm concerned in evolutionary processes, and it is occurring to me that there is a potential here, but maybe not in fact a probability, that it would be possible to dicker with the evolutionary process. And I'd like to know from you, is it possible that by altering genes within a rat or a mouse, that in some way this could in fact become part of the process of meiosis, in which sex cells divide, and that the next generation will inherit the altered genes from this generation, in which case man is creating evolutionary change. What do you think?
Well, the question, I guess, is, are people apt to pass on or transmit these genes? In the type of studies in which we were involved, aimed at treating a lethal anemia, the answer would be clearly no. There was no chance of passing on the genes at all. The principle involved was to take a small amount of bone marrow, which is where blood cells are normally made, and to insert normally functioning genes into bone marrow cells which lack those genes. The bone marrow cells then go back to the patient, and they go back to the bone marrow. If it works, if one is successful, then the only cells in the entire body which have the new genes are the cells of the bone marrow. The genes are nowhere else. They're not in the brain, they're not in the muscle, they're not in the liver, and they're certainly not in the germ cells, in the eggs, for example, or in the sperm. So there is no opportunity for those particular genes ever to be transmitted on. Their only place that they're inserted is in the tissue which is defective, which in this case is the bone marrow.
Father Wasserman.
Doctor, in previous programs we've been dealing with the other guests in this series with ethical questions and with political questions around this whole area. What would you, as a scientist, what restrictions would you like to see on the whole area of genetic engineering, and who do you see as being the responsible persons to put those restrictions on?
Well, there have been many restrictions in the area of genetic engineering going back roughly to about 1973, when the potentialities of doing engineering or gene splicing first became apparent to scientists. At that time, the scientists themselves served as a self-regulatory body, a group of them, and defined, with the help of the federal government, a series of regulations which would cover recombinant DNA or gene splicing. Many of the fears in the original concerns about producing, three-headed guppies and so forth, or let's more realistically say, very virulent viruses that might cause a plague, an Andromeda strain. These fears did not materialize over a long period of observation, and observations involving contact of, I guess, thousands of laboratory workers. Consequently, the scientists and the government, with its scientific advisors, has gradually been relaxing the regulations and the guidelines. I think this type of control has been entirely appropriate. I also think that relaxation of restrictions when the observations and the facts begin to tell you that the hazards may not be as great as one might have thought initially, that's appropriate as well. I guess the simple answer to your question would be government regulatory agencies should be defining the guidelines with the help of both scientific and lay advisors.
Bob Kaler.
I hate to dwell, I hate to dwell on the same subject all the time, but since we're on it, we might as well continue. Part of the reason, as I understand it, that you performed those particular operations in Naples and Jerusalem, those genetic transfer techniques, was the fact that you had applied for permission at UCLA from the Human Subject Protection Committee, and after a long period of 15 months or whatever, they finally ended up denying permission for those same experiments, at which time they already received permission in the other countries. Is there often a problem with the appropriate committees at the various institutions in this country just taking too long to make decisions on things of this sort?
Well, I think the answer to that is yes. But I should clarify one point. My primary reason for doing the initial studies in Naples and Israel was that that's where the patients are who have the disease. The disease affects people mainly who live around the Mediterranean basin. Sicily, Sardinia, Greece, pockets in Israel. This is where the populations who are affected are found, and there tens of thousands of people may be affected. It's relatively rare to find the appropriate patients in this country unless you're dealing with very large communities of Italian or Greek origin. But I guess to come back to your question, may committees take too long? The answer is yes, I think they may. It's often a difficult problem for them, though. The committee may not be comprised of experts in the area. They may have to deal with very unusual proposals or innovative proposals, something new, such as this. They may not themselves have the expertise to deal with it, and they may not necessarily know where to get that expertise. As a generalization, they do the best they can, but the regulations under which they labor may in some cases be excessive. I think this is one reason that many initial trials of new drugs are done not in this country but overseas, simply because it may take years and years before a pharmaceutical company with a new and potentially useful drug could get it tested in this country and get it approved. So it's becoming more and more common, because of these long time delays, for such companies to go abroad.
Don Sprague.
Okay. I'm going to continue along with that line of questioning. We see in movies, Doctor, a competition between scientists to perfect a new surgical technique. What's the competition like in the area of genetic engineering? Did you jump the gun to get a jump on other people doing this type of research? I shouldn't say jump the gun. Did you move ahead with that in mind?
Well, I think scientists are not free of competition. They're like other spheres of our society. And I guess my own opinion would be that's not such a bad thing. I think competition, in some spheres at least, is what makes for progress and advancement. And remember, here, the object of the competition is treating human diseases. Did I jump the gun because of the competitive atmosphere? I would say no, because my colleagues and I were virtually the only, well, we're the only group that I'm aware of that had the expertise both in the gene splicing techniques and in animals, knew how to deal with animal model systems. I think many people were surprised at the rapidity with which these techniques have gone from cells in culture to intact animals. But I don't think we were feeling undue pressure that other scientists were right there. Other scientists were obviously following very important leads, getting important information, which we could use. But I don't...
Richard Snyder.
Dr. Klein, I'm listening with great interest and projecting perhaps into the future within my own mind about possible uses of this. And something that came to mind was, will, in your opinion, just taking a shot at the future, if you will, can you visualize genetic engineering as being used for human cosmetic purposes? A couple of things that came to mind. Genetically engineering out birth defects that are superficial on the skin. Perhaps if a person has enough money, a permanent suntan could be created through alteration of melanocytes, something along this line. Can you visualize this in the future? Perhaps not now.
Well, it's an interesting speculation. I must say I haven't, but it really does sound like science fiction. It would be very hard to do with what we know right now, obviously. I mean, it would be impossible. The technology simply wouldn't exist. The rate of advancement of science, though, is so great that one's predictions about what will be feasible are always off. The answer, honestly, is I don't know. It's an interesting speculation. It sounds a lot like a science fantasy world. It would seem to be a very expensive way to approach that sort of thing when you could go to and much less pleasant than going to Hawaii for your scientific.
Father Wasserman.
Just a short question, Doctor. Could you compare how the American Medical Scientific Society is in relation to genetic engineering, in relation to the scientific community of other countries? Are we the leaders of that? Is it happening elsewhere?
Are we the leaders in the technology of genetic engineering? I would say probably yes, still at this point, but we are not alone in the field. There are many, many countries now in hot pursuit. The Swiss, in collaboration with Americans, have probably one of the finest bioengineering companies going. The Japanese, in their traditional way, are supporting the emergence of new technologies and new industries. And they have at least 2 very well-financed companies supported by the government who are very active, for example, in the production of interferon, an antiviral, maybe an anti-cancer substance. The French are supporting at least 2 major companies devoted really to engineering for mainly agricultural and energy purposes. And the British, as you might suppose, have a sort of nationalized approach. They have a new company called Celgene, I believe, which is supported both by the government and by a consortium of 4 independent companies. Americans are not alone in the field, that's certain, and may not be preeminent for very long.
With that, I'm going to bring our program to conclusion. We're out of time. Dr. Klein, it's been very informative, and we look forward to having you next week when we shall continue this subject.
Thank you.
Ladies and gentlemen, our guest has been Dr. Martin Klein, dealing with genetic engineering as it relates to trying to combat and cure terminal human diseases. I hope you'll be with us again next week when we'll have the same panel to discuss with Dr. Klein this terribly important subject and to bring a conclusion to our 6-week series on this very important subject. I hope you have a very good week. I am Tony Stewart.
The North Idaho College Public Forum can be seen at this same time each week over this station. This production was videotaped earlier by a North Idaho College student crew for viewing at this more appropriate time.